The next speaker is Meike Roskamp from Development Pharma Solutions from BASF, Ludwigshafen, a major excipient manufacturer, talking about Poloxamers, individual, versatile, and safe.
Thank you very much. Thank you for the introduction, and thank you very much for the possibility to talk here today. I'm going to talk about Poloxamers, but actually the real underlying problem I'm going to talk about is the issue of raw material variability for excipients. And even though I'm only going to talk about Poloxamers, I hope that you will be able to see this is maybe something that is also relevant for other excipients that you are currently working with, and that it's maybe worth considering early on in the development. I am part of BASF's Pharma Solution division, where I'm heading the biopharma business, and we provide a broad range of different ingredients for the pharma industry, one of which are the Poloxamers. It's not really moving. Now it's moving. Yes, so Poloxamers. Poloxamers are non-ionic amphiphilic triblock polymers, and after all the talks that have been so far, I think most of you will recognize this type of structure. It has a hydrophobic middle block of polypropylene oxide and two more hydrophilic blocks of polyethylene oxide. The physicochemical properties of these Poloxamers strongly varies with the molecular weight and also the ratio of EO to PO, here highlighted in green and blue. And this dependence of the properties on these parameters makes these materials so versatile, but it also causes some say, variability issues. One property that highly depends on these two parameters is the amphiphilicity of these materials, and with that also the interaction of these Poloxamers with various surfaces like nanosurfaces, but also cells, cell membranes, proteins, and container surfaces like glass and plastic. Poloxamers are around already for quite a while. BASF developed them in the 1950s, and they are used in a wide range of applications, not just pharma. But BASF carries a large range of different pharma-grade Poloxamers that all comply with the Poloxamer monograph and that are available not just in different compositions, but also different final processings. And this helps in order to make them useful for a broad range of pharma applications. And these you can see on the right. And one thing I would like to point out is that the same Poloxamer, let's take P188, is actually used in a range of very different pharmaceutical applications, like plasticizer in tablets, topical formulations, but also the parenteral formulation of antibodies. And for most of these applications, the commercially available pharma-grade material works perfectly fine. So whatever batch you buy, it's going to work in your application. But especially for some new applications, and the biologics field is a perfect example because here it appears quite often. The customers observe lot-to-lot variability. So they see a different performance when they buy a new lot. And while this is, of course, a problem in pharmaceutical applications, it's not completely surprising because actually these materials were, or more precisely, the Poloxamer monograph specifications, and with that the product specifications, were not developed with these new applications in mind. And if you look at some of the specifications, and here is the percent ethylene oxide and the molecular weight, you can see that for a specific Poloxamer, the ranges that you can have in the product specifications are actually quite large. And if you now combine that with the knowledge that these parameters are crucial for the amphiphilicity, it's maybe easy to understand that some applications that are really dependent on these properties show some variability. And these applications are the ones that are interesting candidates for us to develop application-specific poloxamer grades in order to address this variability of performance. And the need for these grades is really driven by increasing quality expectations. On the one hand, based on patient safety concerns. So if you have, for example, an effect on storage stability, or even worse, on efficacy, this is a real problem. And even if the pharma companies fully understand what they need and have internal parameters in addition to the actual product parameters, and then establish a lot selection process, this can still be risky because you never know if the manufacturer actually has a lot in stock that would work for you. So these application-specific grades would take away both of these risks. And how do you now get to such an application-specific grade? So there are two areas that are very useful in reducing variability. One is the absence of performance-critical impurities. So if you know which impurity is important for your application, it's possible to ensure that it's not there or below a certain critical threshold. But the other area is the one I would like to focus on in this talk, and that's a more narrow chemical composition. Because the poloxamers are actually polymers, and with that they are not like a small molecule, one defined chemical structure, but you actually have a range of different chemical structures in this polymer. And for some applications, it's really the combination of these chemical structures that are crucial for performance. And this is something that we would like to address and where we want to produce in a more reproducible way in order to address this variability. And I would like to show you an easy example that shows the concept, and that is the use of these poloxamers in gelling. So poloxamers show a thermoresponsive behavior, which means above a certain concentration and at a certain temperature, these poloxamers form networked gels. It's a reversible process. When you cool them down, they become a liquid again. And this is, for example, an interesting property for in vivo gelling, when you want to do a hydrogel in vivo. But of course, if you want to do that in vivo, it's very important where exactly your gel point is, so the transition temperature when you move from liquid to gel. There are several factors that influence this. One is salt, but another one is actually the molecular weight. And I would like to explain this at the example of two poloxamers that both have the same content of ethylene oxide, so they both have 80% ethylene oxide, but P188 and 338 differ in their molecular weight. The P188 is smaller than the P338. And what you can see here on the graphs, which show the dynamic viscosity versus the temperature, then you always see this jump. That's the gel point when it transitions from liquid to gel. And you can see that the temperature at which that happens is roughly 20 degrees higher for P188 versus the P338. And this is perfectly fine because those are two different materials with different article numbers, so they can have different gel points. But what about the same material and your lot-to-lot variability of the gel point? And I would like to show you the example of P338, which is actually a very interesting poloxamer for this type of application. And what you can see on the left is again the molecular weight that you have as a product specification, which is the monograph specification. And if you look at this, this is actually a quite broad range. So from the lowest to the highest molecular weight, you can have 4,300 Daltons difference. And that for a molecule with an average molecular weight between 12,700 and 17,000 Dalton. On the right, you can see 3 size exclusion chromatograms of different commercial P338 lots. So it shows the molecular weight distribution, which I'm sure everyone here knows. And the blue lot that you can see here has a roughly 1,000 Dalton lower average molecular weight than the two green lots you are seeing. This is not even the maximum you can have. This is a fourth of the difference that you can have from lot to lot. But even this difference already causes a shift in gel point under otherwise identical conditions. So this is a similar graph to what I showed you before. The dynamic viscosity versus the temperature is just zoomed in to the 20 to 40 degree range. And you can see that the blue one gels at a higher temperature compared to these two green lots. For most applications, this is not an issue because you are far away from the gel point or you don't use it in vivo. But for some applications, this variability can actually cause issues. And I think everyone that works with micelles knows this is one phenomenon, but there are others underlying that you could see that would be different between these lots. So this is an example where a more narrow variability for this material would be interesting for someone that wants to develop such an in vivo gelling application. Poloxamers in general are considered to have a low-grade toxicity profile, and both poloxamers that are quite interesting for this gelling application actually are considered to be safe. The P407 at very high concentrations was shown to induce hyperlipidemia and liver toxicity in rodents and rabbits. So if you plan an application with high doses of poloxamer and maybe repeat dosing, it's worth to think about the 338 maybe. The way we go about these application-specific grades, we have our R&D and a central analytical division where we combine the application data with the analytics and then identify what parameters we actually need in these new products. But we highly depend also on customer feedback for this and work closely with some customers in order to make these grades to the requirements of the actual application. And then we also have the production capabilities and expertise to produce these materials with a very high reproducibility and in GMP. But we also recognize that the volumes that we normally produce are not very suitable for these application-specific grades because the annual volumes will be much smaller compared to these poloxamers that are used in a broad range of applications. And also it's actually quite challenging to do that on these very large assets. So we decided to invest into an existing poloxamer manufacturing facility in Wyandotte, which is actually the site where the poloxamers were invented, but where they currently only make technical grade material. And they can not only do poloxamers, they can actually do quite versatile alkoxylations, so various different things. And what we implement there is an even tighter control of the production parameters in order to achieve a very reproducible chemical composition, even beyond molecular weight and % EO, but really the distribution of these polymers. The technical trials are already ongoing, and the technical upgrades, the GMP upgrades, will start in 2024, and we hope that we can have first validation batches of these materials in 2025. And with that, I see I was quite quick. If you have any challenges with poloxamers, please reach out. We are happy to collaborate, or if you're interested in other capabilities, my colleagues are listed here as well. If you want to have some samples, just reach out. Thank you very much.